For today's lab, we will be discussing the preparation of an alpha-beta unsaturated ketone. What exactly is an alpha-beta unsaturated ketone? It's a compound where we see a double bond, or in other words, an unsaturated bond adjacent to a carbonyl group. And this carbon from the double bond that's closest to the carbonyl group, we call it as the alpha carbon. And the second carbon of the double bond, we label it as the beta carbon. You can see there is an alpha-beta unsaturation adjacent to this ketone. That is why we call it an alpha-beta unsaturated ketone. These compounds are also called enones, and they're very useful in the synthesis of complex organic molecules. Unlike ketones, alpha-beta unsaturated ketones have more resonance structures. Usually we see that these electrons shared in the carbon-oxygen double bond can shift towards oxygen and give a lack of electrons or a small positive charge towards this carbon in the carbonyl group. And usually that is where we would see a nucleophile addition occurring. However, since there is a double bond in these alpha-beta unsaturated ketones, the electrons in this double bond can shift towards that carbonyl carbon to account for its lack of electrons, and the positive nature or the lack of electrons would be shifted to that beta carbon position. So now in an alpha-beta unsaturated ketone, the beta carbon is the more susceptible position for a nucleophilic attack to happen. Just to summarize what I discussed in the previous slide, a nucleophile addition to an alpha-beta unsaturated ketone would be at the beta carbon position. And this nucleophilic addition of a nucleophile to the beta carbon, we call it a Michael addition reaction. We're going to look at the reaction mechanism for today's lab step by step. The main reagents that we're using for the experiment today is ethyl acetoacetate, trans-chalcone, which will be the alpha-beta unsaturated ketone, and in addition we'll be using ethanol and a concentrated base solution, which is sodium hydroxide. In the first step of the reaction, this ethyl acetoacetate is deprotonated by a strong base to make a nucleophile, or we also call it a carb anion. The reason that this is possible is because this carbon that is sandwiched between these highly electronegative functional groups is having a electrophilic property with a small positive charge towards it. So in the presence of a strong base, it's more likely to pull the electrons it's sharing with one of these hydrogens and account for its lack of electrons and give away this hydrogen. Molecules that give away protons are usually called acids. So in the presence of a strong base, we would see these hydrogens being lost and a negative charge accumulating around this carbon. Since these hydrogens can be lost this way, they're called as acidic hydrogens or acidic protons. And if you were asked to identify them in a given molecule, you would look for a carbon that is in close proximity to highly electronegative groups, and the hydrogens attached to that carbon would act as acidic protons. By this deprotonation, we now have a nucleophile of from ethyl acetate, and this nucleophile will undertake a nucleophilic addition to trans-chalcone, which is an alpha-beta unsaturated ketone, in order to proceed in this reaction. Now we have the nucleophile that we prepared in the previous slide. It will undertake a nucleophilic attack at the beta carbon position of trans-chalcone and prepare this much more complex molecule. And during that process, the electrons all shift towards this carbonyl oxygen, and the negative charge on this carbonyl oxygen is removed by accepting a proton from water and forming this OH group. Now you can see there's an OH group next to a double bond. These type of molecules, we call them as enols. These enol forms are able to quickly switch to a keto form by a reaction we called tautomerization. So this OH can lose its proton again and shift its electrons back towards the carbon, causing the electrons in these double bonds to shift towards the other carbon, and it's forced to accept a proton. So the proton that was on this oxygen is actually shifted to this carbon over here, and it gets rid of this double bond and forms this ketone group again. And the switch between keto and enol form is tautomerization. Then the next step is an aldol condensation, which is the addition of a carb anion or nucleophile to a carbonyl group, but the special thing about this is it's going to be an intramolecular reaction, and this carbon over here, which initially came from ethyl acetate, is also very electrophilic due to its close proximity to these highly electronegative groups. Therefore, each of these hydrogens can be considered as acidic protons as well, and in the presence of a strong base, one of these hydrogens can be lost, giving a negative property around this carbon, converting it to a carb anion, and that carb anion can undertake a nucleophilic attack on the carbonyl carbon, allowing a new bond to form between this carbon from the carb anion and the carbonyl carbon. So now we have a new bond, and the electrons were shifted to its oxygen, so there is a negative charge around that, which will be taken away by the protonation by ethanol, and finally that OH group will undergo a dehydration by grabbing one of these protons and forming a new carbon-carbon double bond. And you can see that this double bond is adjacent to a carbonyl group. So again, we have an alpha beta unsaturated ketone, but this is much more complex than the reagent molecule that we started with. So this is how we would prepare a much more complex molecule by using an alpha beta unsaturated ketone. Now let's look at the experimental setup for the experiment today. We will have a round bottom flask in which we will have all of our reagents added to it: ethyl acetate, trans-chalcone, ethanol, and concentrated sodium hydroxide solution. And This round bottom flask with all the reagents will be connected to a condenser and also a drying tube. Just to remind you, the purpose of using a condenser is to continuously cool the system while we allow the reaction to proceed for a long time. And this process is known as refluxing. We will be refluxing this reaction for 1 hour, allowing the formation of the final alpha-beta unsaturated ketone product. And that molecule has a yellow color to it. So as the reaction progresses, we will see the solution turn to yellow color. After the reaction is done, we are going to cool this mixture. When we cool a solvent, the solubility of a certain compound in that solvent would decrease. So we would see the precipitation of our product, and we will be able to collect it by vacuum filtration. At this point, this product is still considered a crude product. We would need to isolate our product and purify it by recrystallization in order to obtain a pure sample of the final product. There is a certain impurity which is actually a catalytic compound that comes with trans chalcone that is mixed in the crude product. The first step in the isolation process is to remove that catalytic compound. We do that by transferring the crude product into a centrifuge tube and adding acetone to dissolve the product. The product will dissolve in acetone, but those catalytic compounds will not be soluble in acetone, so it should remain as a solid. By centrifugation, we can pack all that solid to the bottom and then decant the liquid into a 10 ml Erlenmeyer flask. Now we are going to evaporate the acetone. So we're evaporating the solvent, and we will be left with a residue that is our final product, which is still not completely pure, so we still consider as a crude product, and we are going to purify it by recrystallization in ethanol. So this final product is soluble in ethanol when we increase the temperature. So when we heat it, this product will dissolve in ethanol. Any impurities that were trapped in the crude crystals will be released and solubilized in ethanol. Once we allow this mixture to slowly cool back to room temperature, we will see pure crystals forming through selective crystallization, and the impurities should still remain in ethanol. Then by filtration, we should be able to obtain the purified product and then get the actual yield of it and follow characterization of the final product. In the last few experiments, we have been using IR analysis as the method to characterize or confirm our final product. However, we are not able to use IR analysis for this experiment because our final product, the final alpha-beta unsaturated ketone product, has similar functional groups compared to the reagent molecules. Therefore, we won't be able to differentiate between the reagent molecules and the final product. For this reason, we will be using the melting point test, which you guys learned about in OCHEM 1 lab, to confirm whether we got the right product and also get a sense of the purity of the product. The literature value of melting point temperature for this compound is from 111 to 112 Celsius. And if the melting point test derives a melting point temperature range within plus or minus 0.5 to 2 Celsius of this literature value, we can confirm that we obtained the correct product that we were expecting to prepare, and this compound is relatively pure. If the melting point range was much more wider than this and at a lower temperature range, then we can conclude that this sample is not pure and we are not able to confirm whether this is the product that we were expecting to obtain. I will not be discussing about theoretical yield calculation because we have been talking about it in the last few lectures, and the calculation for this experiment is very straightforward. The molar ratio between the two reagents that we measured, that is ethyl acetoacetate and trans-chalcone, is a 1-to-1 ratio, and the molar ratio between them and the final product is also at 1-to-1 ratio. So whatever reagent has the lesser number of moles will serve as the limiting reagent, and you can use that to determine how many moles of the product you could theoretically obtain. That is all for this experiment. Like always, if you have any questions, feel free to contact me through email, and I would see you at office hours. Thank you.